Hello there, and welcome to the Sleepy Science Society. I'm so glad you found your way here tonight. Maybe you've had a long day filled with feelings, or maybe you're simply curious about what happens inside your head when you laugh, cry, feel afraid, or fall in love. Either way, you're in the right place. Tonight's journey is about emotions and the brain, about the beautiful, complex machinery that lets you feel everything you feel. You've got nothing to do but rest. Let your breathing slow and allow yourself to drift as we explore together. And if these gentle explorations help you unwind or give you a quiet way to end your day, consider liking and subscribing to the channel. It helps others discover these peaceful journeys, one curious mind at a time. Now, let's begin. Emotions are perhaps the most human thing about us. They color every moment of our lives, from the smallest irritation when you stub your toe to the overwhelming joy of holding someone you love. They drive our decisions, shape our memories, and connect us to one another in ways that nothing else can. Without emotions, the world would be a flat, colorless place. Music wouldn't move us. Stories wouldn't touch our hearts. We wouldn't cry at weddings or laugh until our sides hurt. Emotions are the texture of consciousness itself. The subjective quality that makes experience worth experiencing. And all of this, every flutter of happiness, every pang of sadness, every surge of anger or moment of peace happens in your brain. Your brain, that 3-pound universe tucked inside your skull, is constantly generating your emotional life. It's not just responding to the world around you, it's interpreting, predicting, and creating your feelings moment by moment. When you see a friend smile, your brain doesn't just register the curved mouth and crinkled eyes. It generates warmth, connection, pleasure. When you hear a sudden, loud noise in the dark, your brain doesn't wait to find out what it is. It floods your body with fear, preparing you to run or fight, all within milliseconds. This happens so quickly, so automatically, that it feels like emotions just happen to you, like they're external forces washing over you. But they're not. They're created inside you, by structures and circuits and chemicals working together in an intricate dance that neuroscientists are only beginning to fully understand. The story of how your brain creates emotions is a story about evolution, about chemistry, about electricity flowing through networks of cells, and about the delicate balance between ancient survival mechanisms and modern cognitive control. It's a story that begins deep in the center of your brain, in a collection of structures so old that they existed long before humans walked the earth. These structures, collectively known as the limbic system, are sometimes called the emotional brain. They're the reason you can feel it all, and they're where our journey begins tonight, in the warm, quiet center of your mind, where feelings are born. The limbic system sits nestled deep within your brain, below the folded outer cortex where you do your conscious thinking, and above the brainstem that keeps your heart beating and your lungs breathing. It's a collection of interconnected structures that work together to generate your emotions, form your memories, and motivate your behavior. The word limbic comes from the Latin word for border, because these structures form a border around the brainstem, creating a ring of tissue that bridges the gap between your most primitive survival functions and your most sophisticated thoughts. Think of the limbic system as the emotional core of your brain, the place where your feelings originate and where they're given their particular flavor and intensity. When you experience an emotion, whether it's joy or fear or sadness or anger, your limbic system is orchestrating that experience, sending signals throughout your brain and body to create the full experience of feeling. It's coordinating with your autonomic nervous system to make your heart race when you're afraid, or to make you feel butterflies in your stomach when you're nervous. It's working with your hormonal system to release the chemicals that sustain emotions over time. And it's communicating with your cortex, the thinking part of your brain, to give your emotions meaning and context. The limbic system is not a single structure but a network, a constellation of brain regions that work in concert. The main players in this system are the hypothalamus, the thalamus, the hippocampus, and the amygdala. Each of these structures has its own specialized role to play, but they're so interconnected, so constantly in communication with one another, that it's sometimes hard to say where one's function ends and another's begins. The hypothalamus, for instance, is a tiny structure, no bigger than an almond, but it's incredibly powerful. It produces hormones, regulates your sleep and wake cycles, controls your body temperature, manages your hunger and thirst, and plays a crucial role in your emotional responses. When you're stressed or afraid, your hypothalamus activates what's called the HPA axis, the hypothalamic pituitary adrenal axis, which releases cortisol and other stress hormones into your bloodstream. When you feel romantic love or maternal affection, your hypothalamus is involved in the release of oxytocin, the bonding hormone that draws you closer to others. The thalamus serves as a relay station for sensory information. Almost everything you see, hear, taste, touch, or feel passes through the thalamus before being sent to other parts of your brain for processing. It's like a switchboard operator, directing incoming signals to the appropriate departments. But the thalamus doesn't just pass information along passively. It filters and modulates sensory input, helping to determine what you pay attention to and how intensely you experience different sensations. This is why, when you're anxious, every little sound seems louder, every shadow seems more menacing. Your thalamus, influenced by your emotional state, is amplifying certain sensory signals and making you hypervigilant. The hippocampus, shaped like a curving seahorse and sitting just next to the amygdala on each side of your brain, is primarily known as the memory center, but its role in emotion is equally important. The hippocampus takes the experiences you have and transforms them into lasting memories. It's the reason you can remember your first day of school, your last birthday party, or what you had for breakfast this morning. But not all memories are created equal. Emotional memories, memories that are tied to strong feelings, are processed differently and stored more robustly than neutral ones. This is because the hippocampus works closely with the amygdala, and when the amygdala signals that an experience is emotionally significant, the hippocampus prioritizes that memory, stamping it with extra importance. This is why you can remember exactly where you were on a day when something emotionally powerful happened, even if you've forgotten countless ordinary days. The emotion, processed by your limbic system, made that memory stick. The hippocampus is also one of the few places in the adult brain where neurogenesis occurs, where new neurons are actually born throughout your life. This process of creating new brain cells in the hippocampus is influenced by your experiences, your stress levels, your exercise habits, and even your emotions. Chronic stress and depression can suppress neurogenesis in the hippocampus, which may be one reason why these conditions are associated with memory problems and difficulty learning new information. But positive experiences, physical activity, and effective emotional regulation can enhance neurogenesis, keeping your hippocampus healthy and your emotional memory sharp. Together, these limbic structures create what we might call your emotional nervous system. They give you the capacity to feel, to remember what you felt, and to use those feelings to guide your behavior. They're ancient in evolutionary terms, present in some form in all mammals and even in many reptiles and birds. Long before our ancestors developed language or complex reasoning, they had limbic systems that helped them survive by generating emotions. Fear helped them avoid predators. Pleasure motivated them to seek food and mates. Sadness helped them grieve losses and maintain social bonds. Anger prepared them to fight threats. These emotions weren't luxuries or optional features of consciousness. They were essential survival tools, and they still are. Even in our modern world, with all its complexity and abstraction, our limbic system continues to do the same job it's always done, generating the feelings that guide us through life. But if we had to choose one structure within the limbic system as the star of the emotional show, it would be the amygdala. This almond-shaped cluster of neurons, sitting deep in each temporal lobe of your brain, is perhaps the single most important structure for processing emotions, especially fear. The amygdala is your brain's alarm system, constantly scanning your environment for potential threats and triggering emotional responses when it detects something dangerous. It's fast, it's powerful, and it operates largely outside of your conscious awareness, making split-second decisions that can save your life. The amygdala is actually not a single structure but a complex of several different nuclei, each with its own specific functions and connections. Neuroscientists typically divide the amygdala into three main groups of nuclei. The basolateral complex, which is the largest part, receives sensory information from the thalamus and cortex and plays a crucial role in learning emotional associations. The central nucleus connects to the brainstem and hypothalamus, and is responsible for generating the physical symptoms of emotions, like increased heart rate, sweating, and the release of stress hormones. And the cortical nuclei connect with the olfactory system, which is why smells can trigger such powerful emotional memories. The scent of a perfume might instantly transport you back to a moment with someone you loved. The smell of smoke might trigger fear and vigilance. This happens because odors have a direct pathway to the amygdala, bypassing the usual relay through the thalamus. The amygdala's primary role, the function that it's best known for, is processing fear. When you encounter something potentially threatening, sensory information travels to your amygdala through two pathways. The fast pathway, sometimes called the low road, goes directly from your thalamus to your amygdala, bypassing your cortex entirely. This pathway is crude and imprecise, but it's incredibly fast, allowing you to react to danger in a fraction of a second. If you're walking in the woods and you see something long and curved on the path ahead, The low road might trigger a fear response before you've even consciously registered what you're seeing. Your amygdala sounds the alarm. Your body floods with adrenaline, and you jump back, all before your cortex has had time to analyze the situation and realize that what you saw was just a curved stick, not a snake. The slow pathway, or high road, takes a bit longer. Information goes from your thalamus to your visual cortex, where it's carefully analyzed, and then to your amygdala. This pathway gives you more accurate information. It's the cortex telling your amygdala, "Don't worry, it's just a stick, not a snake." In most situations, the high road eventually overrides the low road, allowing you to calm down once you realize there's no real danger. But in that brief moment before your cortex catches up, your amygdala has already prepared your body to run or fight. This system evolved because in a world full of predators, it was better to jump at 10 sticks if it meant you'd avoid one real snake. False alarms were worth it if they kept you alive. When the amygdala detects a threat, it sets off a cascade of responses throughout your brain and body. It activates the sympathetic nervous system, triggering the famous fight-or-flight response. Your heart races, pumping blood to your muscles. Your breathing quickens, bringing more oxygen into your lungs. Your pupils dilate, sharpening your vision. Glucose is released into your bloodstream, giving you a burst of energy. Meanwhile, non-essential functions like digestion slow down. Blood is redirected away from your skin and digestive organs and toward your large muscles. Your amygdala has essentially put your body on high alert, preparing you for action. At the same time, the amygdala sends signals to your hypothalamus, which activates the HPA axis, releasing cortisol and other stress hormones that sustain the fear response over longer periods. And it communicates with your frontal cortex, particularly the anterior cingulate cortex and the orbitofrontal cortex, regions involved in conscious emotional experience and decision-making. But the amygdala isn't just about fear. Research has shown that it also plays important roles in processing other emotions, including pleasure, reward, and social interactions. Different parts of the amygdala respond to different emotional stimuli. The left amygdala, for instance, seems to be particularly involved in positive emotions and reward processing, while the right amygdala is more associated with negative emotions like fear and sadness. Electrical stimulation of different parts of the amygdala can produce dramatically different emotional experiences. Stimulation of some regions produces intense fear or anxiety, while stimulation of others can produce feelings of pleasure or even euphoria. This suggests that the amygdala is a more nuanced structure than we once thought. Not just a fear center, but a general emotional evaluation system that assigns emotional significance to stimuli, whether positive or negative. The amygdala is also crucial for emotional learning, particularly fear conditioning. This is the process by which we learn to associate neutral stimuli with danger. The classic example is Pavlov's dogs, who learned to associate the sound of a bell with food and began to salivate at the sound alone. The amygdala mediates a similar process with fear. If you hear a loud noise and then immediately feel pain, your amygdala will create an association between that noise and danger. The next time you hear that noise, your amygdala will trigger a fear response, even if there's no pain this time. This is how phobias develop, how people who've been in car accidents become afraid of driving, how survivors of trauma can be triggered by seemingly harmless reminders of their experience. The amygdala has learned that certain cues predict danger, and it responds accordingly, trying to protect you. What happens when the amygdala is damaged or doesn't function properly? There are rare cases of people with bilateral amygdala damage, where both the left and right amygdala have been destroyed by disease. The most famous case is a woman known as patient SM, who has Urbach-Wiethe disease, a condition that caused calcium deposits to destroy her amygdalae. Patient SM has a profound inability to experience fear. She can't recognize fearful expressions on other people's faces. She doesn't avoid dangerous situations that would terrify most people. She reports never feeling afraid, even in situations that would normally trigger intense fear responses. In experiments, researchers have tried everything from showing her scary movies to taking her to a haunted house to exposing her to snakes and spiders, creatures she says she dislikes. Yet she doesn't experience the physical sensations of fear, the racing heart and sweaty palms that normally accompany threatening situations. Her life is marked by a strange fearlessness that, while it might sound appealing, has actually put her in danger many times, because fear, for all its unpleasantness, serves a crucial protective function. Interestingly, people with amygdala damage can sometimes still experience fear in certain contexts, particularly if the threat is internal rather than external. Patient SM, for instance, reported feelings of panic when researchers had her inhale carbon dioxide, which creates a sensation of suffocation. This suggests that there are multiple fear systems in the brain, and the amygdala is primarily involved in processing external threats detected through the senses, while other brain regions may be responsible for responding to internal bodily states. It's a reminder that the brain's emotional systems are redundant and distributed, not localized to single structures. The amygdala's size and activity also vary between individuals, and these differences may influence personality and susceptibility to emotional disorders. People with anxiety disorders often show heightened amygdala activity, particularly in response to uncertain or ambiguous stimuli. Their amygdalae are essentially overactive, detecting threats where none exist and generating excessive fear responses. Children who are behaviorally inhibited, meaning they're shy and fearful in new situations, tend to have larger left amygdalae. And people who've experienced severe trauma, particularly childhood trauma, may have altered amygdala function, which can contribute to post-traumatic stress disorder. Understanding the amygdala's role in emotion has therefore been crucial, not just for basic neuroscience, but also for understanding and treating mental health conditions. Working alongside the amygdala, the hippocampus plays a vital but different role in emotional life. While the amygdala is evaluating the emotional significance of experiences and triggering emotional responses, the hippocampus is busy encoding those experiences into memory. These two structures sit right next to each other in the temporal lobe, and they're intimately connected, constantly exchanging information. When something emotionally important happens, both structures are activated together, which is why emotional experiences create such strong, vivid memories. The hippocampus gets its name from its resemblance to a seahorse when viewed in cross-section. There are two hippocampi, one in each hemisphere, and together they form a crucial part of the brain's memory system. The hippocampus is particularly important for episodic memory, your memory of specific events and experiences. It's what allows you to remember what happened yesterday, to navigate through your neighborhood, to recall the face of a friend you haven't seen in years. Without a functioning hippocampus, you would lose the ability to form new long-term memories, a condition known as anterograde amnesia. You would be stuck in an eternal present, unable to learn new information or remember anything that happens to you. But the hippocampus doesn't just store memories indiscriminately. It works together with the amygdala to prioritize emotional memories. When the amygdala signals that an experience is emotionally arousing, whether positively or negatively, it enhances the encoding and consolidation of that memory in the hippocampus. This makes evolutionary sense. It's more important to remember events that have emotional significance, things that might affect your survival or well-being, than to remember neutral, mundane events. You're more likely to remember your wedding day than a random Tuesday afternoon 2 years ago, not because your brain has limited storage space, but because emotional significance serves as a kind of biological highlighter, marking certain memories as important. This enhancement of emotional memories happens through several mechanisms. The amygdala releases neurotransmitters and hormones, particularly norepinephrine and cortisol, that enhance synaptic plasticity in the hippocampus. Synaptic plasticity is the brain's ability to strengthen or weaken connections between neurons, and it's the cellular basis of learning and memory. When you experience something emotionally significant, the flood of neurotransmitters from the amygdala makes the synaptic connections in your hippocampus more plastic, more likely to change and strengthen. This means the memory of that event gets encoded more deeply, more permanently. It's why you can remember traumatic events in vivid detail years later, sometimes with an intensity that feels almost like reliving the event. The emotional arousal at the time stamped that memory deep into your brain. The hippocampus is also involved in contextual memory, your memory of the circumstances and environment in which events occur. When you remember a specific moment from your past, you don't just remember what happened. You remember where you were, what time of day it was, what the weather was like, who else was there. These contextual details are processed and stored by the hippocampus, and they're crucial for understanding and using your memories. Context helps you distinguish between similar events and allows you to recall appropriate responses. If you were once bitten by a dog in a park, your amygdala might create a fear response to dogs, but your hippocampus stores the context, helping you understand that the dangerous event happened in a specific place at a specific time. This contextual information allows you to be cautious around dogs in parks while still being comfortable with your friend's gentle dog at home. The hippocampus is also one of the most plastic, adaptable parts of the brain. As mentioned earlier, it's one of the few brain regions where new neurons continue to be born throughout adulthood, a process called adult neurogenesis. The rate of neurogenesis in the hippocampus is influenced by many factors. Exercise increases neurogenesis, which is one reason why physical activity is so good for memory and mood. Enriched environments, with lots of opportunities for learning and exploration, also enhance neurogenesis. On the flip side, chronic stress suppresses neurogenesis, which may contribute to the memory problems and cognitive difficulties that often accompany depression and anxiety. The stress hormone cortisol, when present in high levels over long periods, can actually damage hippocampal neurons and shrink the hippocampus. Brain imaging studies have found that people with chronic depression often have smaller hippocampi than healthy individuals, and the degree of shrinkage correlates with the duration and severity of depression. This relationship between stress, the hippocampus, and emotion creates a kind of vicious cycle. Chronic stress shrinks the hippocampus, which impairs emotional regulation and makes it harder to cope with stress, which leads to more stress, further damaging the hippocampus. Breaking this cycle is one of the goals of treatments for depression and anxiety. Antidepressant medications, particularly selective serotonin reuptake inhibitors, have been shown to promote neurogenesis in the hippocampus. Psychotherapy, especially cognitive behavioral therapy, can help patients develop better coping strategies and reduce chronic stress. Exercise, mindfulness meditation, and social support all appear to have protective effects on the hippocampus. These interventions don't just make people feel better psychologically, they actually promote structural changes in the brain, rebuilding the hippocampus and restoring its function. The interplay between the amygdala and hippocampus creates the full texture of emotional memory. The amygdala provides the emotional intensity, the visceral feeling, while the hippocampus provides the narrative context, the who, what, where, and when. Together, they create memories that are not just factual records, but emotionally charged stories. This is why, when you remember falling in love, you don't just recall the facts of the matter. You remember the feeling of butterflies in your stomach, the way your heart raced, the particular slant of light in the room, the song that was playing. The memory is rich and multidimensional because it was encoded by multiple brain systems working together. But emotions aren't just generated and remembered. They're also regulated. And this is where the prefrontal cortex comes in. If the limbic system is the emotional core of your brain, then the prefrontal cortex is the regulatory system, the part of your brain that can modulate, suppress, or reinterpret emotional responses. The prefrontal cortex, located right behind your forehead in the frontal lobes, is the most evolutionarily recent part of the human brain. It's what makes us distinctly human, giving us the capacity for abstract reasoning, planning, decision-making, and self-control. And crucially, it gives us the ability to regulate our emotions. The prefrontal cortex is connected to the limbic system through extensive neural pathways. It receives emotional information from the amygdala and other limbic structures, and it sends regulatory signals back to them. Think of it as a conversation between two parts of your brain. Your amygdala might be saying, "This is scary, we need to run," while your prefrontal cortex is saying, "Hold on, let's think about this rationally. Is this really dangerous?" This conversation happens constantly, every time you experience an emotion, and the balance between these two systems determines how you feel and how you behave. One of the key regions within the prefrontal cortex for emotion regulation is the ventromedial prefrontal cortex, or VMPFC. This region is involved in assigning value and emotional meaning to experiences. It helps you evaluate whether something is good or bad, rewarding or punishing. People with damage to the VMPFC often have difficulty making decisions, particularly decisions that involve weighing emotional factors. They might understand intellectually what the right choice is, but they can't feel which option is better, and this absence of emotional guidance leaves them paralyzed, unable to commit to a decision. This shows that emotions aren't just irrational forces that need to be suppressed. They're essential guides that help you navigate the world, and the VMPFC is where emotional and rational information come together to inform your choices. The anterior cingulate cortex, or ACC, is another crucial prefrontal region for emotion. It's involved in detecting conflicts and errors, monitoring your emotional state, and regulating attention. When you experience conflicting emotions, or when your emotional response conflicts with your goals, the ACC is activated. It helps you notice when something isn't right emotionally and signals the need to adjust your response. The ACC is also involved in empathy and social pain. Brain imaging studies have shown that when people watch someone they care about in pain, or when they experience social rejection, the ACC lights up. Social pain, it turns out, activates similar brain regions as physical pain, which is why heartbreak and loneliness can hurt just as much as a physical injury. The dorsolateral prefrontal cortex, or DLPFC, is involved in working memory, cognitive control, and the conscious regulation of emotion. When you deliberately try to reinterpret a situation to change how you feel about it, a process called cognitive reappraisal, your DLPFC is working hard. For instance, if you're nervous about giving a presentation, you might reframe your anxiety as excitement, telling yourself that the butterflies in your stomach are a sign that you're energized and ready, not that you're about to fail. This reappraisal involves your DLPFC exerting top-down control over your amygdala, dampening the fear response and shifting your emotional experience. Research has shown that people who are good at cognitive reappraisal, who can successfully reframe situations to regulate their emotions, show increased activity in the DLPFC and decreased activity in the amygdala. They're literally using their thinking brain to calm down their emotional brain. The development of the prefrontal cortex has important implications for emotional regulation across the lifespan. The prefrontal cortex is one of the last brain regions to fully mature, continuing to develop well into a person's 20s. This is why children and adolescents often have difficulty regulating their emotions. Their limbic systems, particularly their amygdala, are fully developed and highly reactive, but their prefrontal cortices are still maturing, leaving them with strong emotional responses and relatively weak regulatory control. This developmental mismatch is why teenagers can be so moody and impulsive. They feel things intensely, but they don't yet have the full neural machinery to modulate those feelings. As the prefrontal cortex develops, emotional regulation improves, and people generally become better at managing their emotions as they move into adulthood. But the prefrontal cortex isn't just about suppressing emotions. It's also about generating them in more sophisticated ways. The emotional experiences of an adult are richer and more nuanced than those of a child, not just because adults have had more experiences, but because the prefrontal cortex adds layers of meaning and interpretation. You don't just feel happy or sad. You feel wistful, nostalgic, bittersweet, content, elated, melancholy, and 1,000 other subtle shades of emotion. These complex feelings emerge from the interplay between your limbic system, which generates basic emotional responses, and your cortex, which interprets and elaborates on those responses based on your memories, beliefs, and goals. The regulatory relationship between the prefrontal cortex and the amygdala can be disrupted in various ways, leading to emotional disorders. In depression, the prefrontal cortex often shows reduced activity, while the amygdala may be hyperactive. This imbalance means that negative emotions are generated strongly but aren't adequately regulated, leading to persistent sadness and difficulty experiencing positive emotions. In anxiety disorders, the prefrontal cortex may struggle to inhibit an overactive amygdala, resulting in excessive fear and worry. Many therapeutic interventions, from medication to psychotherapy to mindfulness training, work by helping to restore the balance between these systems, strengthening prefrontal control and calming limbic reactivity. All of these emotional processes, from the rapid threat detection of the amygdala to the memory encoding of the hippocampus to the regulatory control of the prefrontal cortex, depend on neurotransmitters, the chemical messengers that allow neurons to communicate. Neurotransmitters are molecules released by one neuron that travel across the tiny gap between neurons, called a synapse, and bind to receptors on the next neuron, either exciting it and making it more likely to fire, or inhibiting it and making it less likely to fire. Different neurotransmitters have different effects on mood and emotion, and the balance of these chemicals in your brain profoundly influences how you feel. Dopamine is perhaps the most famous neurotransmitter when it comes to emotion, often called the feel-good chemical or the reward neurotransmitter. Dopamine is produced in several areas of the brain, including the substantia nigra and the ventral tegmental area, and it's released in key brain regions including the prefrontal cortex, the striatum, and the nucleus accumbens. Dopamine plays a crucial role in motivation, reward, and pleasure. When you achieve a goal, eat something delicious, or receive a compliment, your brain releases dopamine, creating a feeling of satisfaction and pleasure. This dopamine release reinforces the behavior that led to the reward, making you more likely to repeat it in the future. This is the basis of the brain's reward system, a fundamental mechanism that drives learning and motivation. But dopamine is more complex than just a simple pleasure signal. It's also involved in wanting and motivation, in the anticipation of reward rather than just the reward itself. Some research suggests that dopamine is released most strongly not when you receive a reward, but when you expect a reward. The excitement you feel when you're about to open a gift, the anticipation before a first date, the motivation to work hard for a promotion, all of these involve dopamine. In this view, dopamine is less about liking and more about wanting, about the drive to seek out rewarding experiences. This distinction is important for understanding addiction, where dopamine plays a central role. Drugs of abuse cause massive releases of dopamine, far beyond what natural rewards produce, and this overwhelming dopamine signal hijacks the brain's reward system. The addict's brain learns that the drug is the ultimate reward, and the desire for the drug, driven by dopamine, becomes overpowering, even as the actual pleasure from the drug diminishes over time. Dopamine also interacts with the prefrontal cortex to support cognitive functions like working memory, attention, and cognitive flexibility. Optimal levels of dopamine in the prefrontal cortex improve your ability to hold information in mind, switch between tasks, and regulate your behavior. Too little dopamine, as seen in conditions like Parkinson's disease or attention deficit hyperactivity disorder, can cause problems with motivation, attention, and emotional flatness. Too much dopamine, as may occur in schizophrenia or with the use of stimulant drugs, can cause hyperactivity, impulsivity, and, in severe cases, psychosis. The relationship between dopamine and prefrontal function follows what's called an inverted U-shaped curve. Moderate levels are optimal, while both too little and too much impair function. Serotonin is another crucial neurotransmitter for emotion, often associated with mood regulation and feelings of contentment and well-being. Serotonin is produced in the raphe nuclei, clusters of neurons in the brainstem, and it's distributed widely throughout the brain, including to the prefrontal cortex, the limbic system, and many other regions. Serotonin has a calming, stabilizing effect on mood. It helps you feel content, patient, and emotionally balanced. Low levels of serotonin are strongly associated with depression, anxiety, and impulsive behavior. This is why many antidepressant medications are selective serotonin reuptake inhibitors, or SSRIs. These drugs work by increasing the amount of serotonin available in the synapses between neurons, which over time can improve mood and reduce anxiety. Serotonin also plays an important role in regulating other neurotransmitters, particularly dopamine. There's a reciprocal relationship between the serotonin and dopamine systems. Serotonin tends to inhibit dopamine, providing a kind of brake on reward-seeking and impulsive behavior. When serotonin levels are low, dopamine can become dysregulated, leading to increased impulsivity, aggression, and difficulty controlling urges. This is why low serotonin is associated not just with depression, but also with impulsive aggression and poor impulse control. Serotonin helps you pause, think through consequences, and resist immediate temptations in favor of longer-term goals. It's essential for what psychologists call self-regulation, the ability to control your behavior in accordance with your values and goals, rather than just acting on your immediate feelings and impulses. Interestingly, serotonin is not only found in the brain. In fact, the vast majority of your body's serotonin, around 90%, is produced in your gut by cells in the intestinal lining. This gut serotonin doesn't cross into the brain, but it influences digestion, gut motility, and the gut-brain axis, the bidirectional communication between your gastrointestinal system and your central nervous system. This is why emotional states can affect digestion, and why digestive problems can influence mood. The connection between the gut and the brain, mediated in part by serotonin, is an area of intense research interest, with implications for understanding depression, anxiety, and even autism spectrum disorders. Norepinephrine, also known as noradrenaline, is another monoamine neurotransmitter involved in emotion and arousal. It's produced primarily in the locus coeruleus, a small nucleus in the brainstem, and it's released throughout the brain in response to novel, stressful, or emotionally arousing stimuli. Norepinephrine increases alertness, attention, and arousal. It's part of the fight-or-flight response, preparing your brain and body for action. When your amygdala detects a threat, it triggers the release of norepinephrine, which sharpens your focus, increases your heart rate, and makes you more vigilant. This heightened state of arousal helps you respond quickly to danger, but it can also contribute to anxiety if it's prolonged or if it occurs in situations where there's no real threat. Norepinephrine also plays a role in memory, particularly in the encoding of emotionally arousing memories. When something exciting or stressful happens, norepinephrine is released in the amygdala and hippocampus, where it enhances memory consolidation. This is why you remember emotionally charged events so vividly. The norepinephrine released during the experience acts like a memory enhancer, making those memories stick. Medications that block norepinephrine, called beta blockers, have been explored as treatments for preventing post-traumatic stress disorder. The idea is that if you take a beta blocker soon after a traumatic event, it might reduce the norepinephrine-mediated enhancement of the traumatic memory, making the memory less vivid and less likely to cause long-lasting distress. The balance between these three monoamines, dopamine, serotonin, and norepinephrine, is crucial for emotional health. They don't work in isolation but constantly interact, modulating each other's effects. Dopamine and norepinephrine tend to increase arousal, attention, and motivation, while serotonin provides a calming, inhibitory influence. When these systems are in balance, you feel emotionally stable, motivated, and able to cope with stress. When they're out of balance, emotional problems can arise. Too much dopamine and norepinephrine relative to serotonin might lead to anxiety, impulsivity, and mania. Too little dopamine and too much serotonin might lead to emotional flatness and lack of motivation. Too little of all three might contribute to the profound lack of energy and pleasure seen in severe depression. Other neurotransmitters also play important roles in emotion. GABA, or gamma-aminobutyric acid, is the brain's primary inhibitory neurotransmitter. It reduces neuronal activity and has a calming effect. Anti-anxiety medications like benzodiazepines work by enhancing GABA activity, which is why they produce relaxation and sedation. Glutamate is the primary excitatory neurotransmitter, and it's essential for learning and memory, including emotional learning. Endorphins, the brain's natural opioids, are released during exercise, laughter, and other pleasurable activities, producing feelings of euphoria and reducing pain. Oxytocin, sometimes called the love hormone, is released during social bonding, particularly during physical touch, sex, and childbirth. It promotes feelings of trust, attachment, and connection, and it plays a crucial role in maternal behavior and romantic love. The intricate interplay of all these chemicals, working within the complex circuits of your limbic system and cortex, creates the full spectrum of your emotional life. And different emotions involve different patterns of brain activity and different neurochemical profiles. When neuroscientists use brain imaging techniques like functional MRI to study emotions, they've found that different emotions are associated with distinct patterns of activation across the brain. These patterns are complex and distributed, involving many regions working together, but there are some consistencies that have emerged from research. Happiness tends to activate several regions, including the prefrontal cortex, particularly on the right side, the left amygdala, the precuneus, and the insula. The activation of frontal regions suggests that happiness involves not just feeling good, but also awareness of that feeling and the ability to maintain it. The left amygdala's involvement in positive emotions may seem surprising, given that the amygdala is best known for processing fear. But it highlights that the amygdala is a general emotional processing center, not just a fear center. Happiness also involves the brain's reward system, including the ventral striatum and nucleus accumbens, regions rich in dopamine that respond to rewarding stimuli. Fear, as we've discussed extensively, strongly activates the amygdala, particularly the right amygdala, along with the hypothalamus and regions of the left frontal cortex. The amygdala's response to fear is rapid and often occurs before you're consciously aware of what you're afraid of. The hypothalamus's activation triggers the physiological components of fear, the racing heart and rapid breathing. And the frontal cortex becomes involved as you try to make sense of what's happening and decide how to respond. In situations of chronic fear or anxiety, the amygdala may remain chronically hyperactive, keeping you in a constant state of vigilance, even when there's no immediate threat. Sadness is associated with increased activity in the occipital lobe, the left insula, the left thalamus, the amygdala, and the hippocampus. The involvement of the hippocampus makes sense given that sadness often involves reminiscing, thinking about loss, remembering better times. The insula, a region deep within the cortex that processes bodily sensations and integrates them with emotions, becomes active during sadness, which may relate to the physical sensations of sadness, the heaviness in your chest, the lump in your throat. Sadness also often involves reduced activity in certain frontal regions, particularly those involved in motivation and reward. This is why, when you're sad, it can be hard to feel motivated or to find pleasure in activities you usually enjoy. Anger activates the right hippocampus, the amygdala, both sides of the prefrontal cortex, and the insular cortex. The amygdala's involvement reflects anger's role as a response to threat or frustration. Unlike fear, which prepares you to escape, anger prepares you to confront and fight. The prefrontal cortex's activation during anger is interesting because it suggests that anger, while it feels like a hot, uncontrollable emotion, actually involves significant cognitive processing. You're not just feeling angry. You're thinking about what made you angry, assigning blame, considering responses. This is why cognitive reappraisal can be so effective for managing anger. By changing how you think about the situation, by reframing it or considering alternative explanations, you can often diffuse your anger. Surprise activates the bilateral inferior frontal gyrus and the bilateral hippocampus. The hippocampus's involvement makes sense because surprise occurs when something doesn't match your expectations, when there's a mismatch between what you predicted and what actually happened. Your brain has to quickly update its model of the world, and the hippocampus is involved in that updating. Surprise can be pleasant or unpleasant depending on whether the unexpected event is positive or negative. Pleasant surprise, like an unexpected gift, activates reward regions and can quickly transition into happiness. Unpleasant surprise, like hearing bad news, can quickly transition into fear or sadness. Surprise is in some ways the most cognitively driven emotion because it depends entirely on your expectations and predictions. Disgust primarily activates the insula and parts of the basal ganglia. The insula is particularly interesting here because it's involved in processing visceral sensations, the feelings in your body. Disgust often involves a strong visceral reaction. Nausea, the urge to vomit or pull away. This makes sense evolutionarily because disgust originally evolved to protect us from contamination and disease. Foods that look or smell spoiled trigger disgust, motivating us to avoid eating them. But disgust has expanded beyond physical contamination to include moral disgust. The feeling you get when you encounter behavior you find morally reprehensible. Brain imaging studies have shown that moral disgust activates many of the same regions as physical disgust, suggesting that we use the same neural systems to evaluate both physical and moral purity. These patterns of brain activation show that emotions are not simple, localized phenomena. They're complex, distributed states involving many brain regions working in concert. And importantly, emotions don't have fixed, universal patterns. The same emotion can be experienced differently by different people or in different contexts. And these differences are reflected in varying patterns of brain activity. This has led some neuroscientists to question the traditional view that there are a small number of basic, universal emotions. Instead, they propose that emotions are constructed by the brain on the fly, assembled from more basic components based on your past experiences, your current situation, and your body's state. In this view, emotions are predictions that your brain makes about what your bodily sensations mean in a given context. This constructionist view is still debated, but it highlights the incredible flexibility and context sensitivity of emotional experience. The relationship between emotions and the brain is also powerfully influenced by stress. Stress is not an emotion in itself, but rather a physiological and psychological state that occurs when you perceive that demands exceed your ability to cope. Stress triggers a coordinated response involving the hypothalamus, the pituitary gland, and the adrenal glands, collectively known as the HPA axis. When you encounter a stressor, your hypothalamus releases corticotropin-releasing hormone, which signals the pituitary gland to release adrenocorticotropic hormone, which in turn signals the adrenal glands to release cortisol and other stress hormones. Cortisol has wide-ranging effects throughout the body, increasing blood sugar, suppressing the immune system, and altering brain function. In the short term, the stress response is adaptive. It mobilizes energy, sharpens attention, and prepares you to deal with challenges. But when stressor becomes chronic, when you're constantly activating this system without adequate recovery, it can have damaging effects on the brain and on emotional health. Chronic stress shrinks the hippocampus, impairing memory and emotional regulation. It hypertrophies the amygdala, making it larger and more reactive, which increases anxiety and fear. And it impairs the functioning of the prefrontal cortex, reducing your ability to regulate emotions, make good decisions, and think clearly. These structural and functional changes can create a vicious cycle where stress impairs the very brain regions you need to cope with stress, making you more vulnerable to further stress and to the development of depression and anxiety disorders. Chronic stress also disrupts the balance of neurotransmitters. It can deplete serotonin and dopamine, contributing to low mood and lack of motivation. It can increase norepinephrine, contributing to anxiety and hyperarousal. And it can lead to a state called learned helplessness, where repeated exposure to uncontrollable stress makes you feel powerless and unable to change your situation, even when opportunities for change arise. This learned helplessness is thought to be a core feature of depression, and it's associated with changes in how the brain processes reward and motivation. But the brain is not defenseless against stress. It has resilience mechanisms, ways of protecting itself and bouncing back from adversity. One of the most important factors in resilience is social support. Feeling connected to others, having people you can rely on, activates brain systems that buffer against stress. Oxytocin, released during positive social interactions, has anti-stress effects. It reduces activity in the amygdala and HPA axis, promoting feelings of calm and safety. This is why being with loved ones can make you feel better when you're stressed. It's not just emotional comfort. It's a biological process that's dampening your stress response at the level of your brain and hormones. Exercise is another powerful resilience factor. Physical activity increases neurogenesis in the hippocampus, promotes the release of brain-derived neurotrophic factor, a protein that supports the growth and survival of neurons, and increases levels of endorphins and other mood-enhancing chemicals. Regular exercise has been shown to be as effective as antidepressant medication for mild to moderate depression, and it's a valuable component of treatment for anxiety disorders. Mindfulness meditation, the practice of paying attention to the present moment without judgment, has also been shown to have beneficial effects on the brain. Regular meditation can increase the thickness of the prefrontal cortex, enhance connectivity between the prefrontal cortex and the amygdala, and reduce amygdala reactivity. People who meditate regularly often report feeling more emotionally balanced and better able to cope with stress. These interventions, social connection, exercise, meditation, work in part by engaging the brain's emotion regulation systems. They strengthen the prefrontal cortex's ability to modulate the limbic system. They support the health of the hippocampus, and they help maintain a healthy balance of neurotransmitters. Understanding the neuroscience of stress and resilience gives us insight into why these practices are so beneficial and provides a biological rationale for incorporating them into mental health treatment. The process by which we learn emotional responses, and by which those responses can be modified, is called emotional conditioning, and it's one of the most well-studied phenomena in behavioral neuroscience. The principles of emotional conditioning were first described by Ivan Pavlov in his famous experiments with dogs. Pavlov showed that if a neutral stimulus, like the sound of a bell, is repeatedly paired with a stimulus that naturally triggers a response, like food, eventually the neutral stimulus alone will trigger the response. The dogs learned to associate the bell with food and began to salivate at the sound of the bell even when no food was present. This is classical conditioning, and it applies to emotions just as much as to salivation. Fear conditioning works on the same principles. If a neutral stimulus, like a tone or a light, is paired with something aversive, like a mild electric shock, you'll quickly learn to associate the tone with the shock. After just a few pairings, the tone alone will trigger a fear response, increased heart rate, sweating, a startle reaction, even though there's no shock. Your amygdala has learned that the tone predicts danger, and it's preparing your body to respond. This kind of learning is incredibly fast and powerful, which makes sense evolutionarily. If a rustling in the grass was once followed by a snake, you'd better learn to fear that rustling quickly, because next time it might save your life. But once an emotional association is learned, how do you unlearn it? This is a crucial question, because many emotional problems involve learned associations that are no longer adaptive. Someone who was in a car accident might develop a fear of driving. Someone who was bitten by a dog might become afraid of all dogs. Someone who experienced a panic attack in a crowded place might start avoiding crowds. These fears are based on real experiences, but they often generalize too broadly and persist long after the danger has passed. How does the brain overcome learned fear? The answer is a process called extinction. Extinction doesn't erase the original fear memory. Instead, it involves learning a new, competing memory. If you're afraid of dogs because you were once bitten, exposure therapy would involve gradually, safely encountering dogs without being bitten. Over time, your brain learns a new association. Dogs are not dangerous, and this new learning competes with and eventually overshadows the old fear memory. At the neural level, extinction involves the prefrontal cortex, particularly the ventromedial prefrontal cortex, inhibiting the amygdala's fear response. During extinction training, cells in the VMPFC that inhibit the amygdala become more active, and over time, this inhibitory connection strengthens. The fear memory is still there in your amygdala, but the VMPFC is actively suppressing it. This explains why extinction is context-dependent. If you overcome your fear of dogs in one setting, say a therapist's office, the fear might return when you encounter a dog in a different setting, like a park. The extinction learning was specific to the context where it occurred, and in a new context, the original fear memory can resurface. This phenomenon, called renewal, is a challenge for treating anxiety disorders and phobias. It means that extinction training needs to occur in multiple contexts to be fully effective, and it means that even after successful treatment, there's always some risk of the fear returning under certain circumstances. Understanding the neural mechanisms of fear, learning, and extinction has led to improvements in exposure therapy, the most effective treatment for anxiety disorders and phobias. Modern exposure therapy doesn't just involve confronting your fears. It involves doing so in a way that maximizes extinction learning, engaging the prefrontal cortex and strengthening its inhibitory control over the amygdala. Techniques like cognitive reappraisal, where you consciously reframe the feared situation, enhance this prefrontal engagement. Mindfulness-based approaches, where you observe your fear without judgment or avoidance, also seem to promote extinction by allowing you to experience the fear response without reinforcing it. Emotions don't exist in a vacuum. They're fundamentally social. Much of your emotional life is shaped by your interactions with other people, and your brain has specialized systems for processing social and emotional information about others. One of the most fascinating discoveries in neuroscience is the existence of mirror neurons. Neurons that fire both when you perform an action and when you observe someone else performing the same action. Mirror neurons were first discovered in the motor cortex of macaque monkeys. Researchers found that certain neurons would fire when the monkey reached for a piece of food. But remarkably, the same neurons would also fire when the monkey watched the researcher reach for food. The neuron was mirroring the observed action, as if the monkey were performing it itself. In humans, mirror neuron systems have been found not just for actions, but for emotions as well. When you see someone smile, neural circuits that are active when you yourself smile become active. When you see someone in pain, regions that process your own pain, including the anterior cingulate cortex and the insula, become active. This neural mirroring is thought to be the basis of empathy, your ability to understand and share the feelings of others. You don't just intellectually deduce what someone else is feeling. You simulate their emotional state in your own brain, creating a kind of internal resonance with their experience. This empathic resonance is automatic and largely unconscious. You can't help but wince when you see someone stub their toe or feel a flutter of happiness when you see a friend's joyful expression. This automatic emotional contagion is the foundation of social connection. It's what allows parents to attune to their infants' needs, friends to comfort each other, and communities to come together in shared grief or celebration. But empathy is not just automatic mirroring. It also involves more cognitive components, like perspective taking and emotional regulation. You can feel what someone else is feeling, but you also maintain awareness that it's their feeling, not yours. This distinction is important, because without it, you'd be overwhelmed by others' emotions, unable to help because you're too caught up in the feeling yourself. The brain systems involved in empathy overlap significantly with those involved in processing your own emotions. The amygdala, the insula, the anterior cingulate cortex, and various prefrontal regions are all active during empathic responses, but there are also differences. When you experience your own pain, certain sensory regions that process the physical sensation of pain are active. When you observe someone else in pain, those sensory regions are not active, but the emotional and evaluative components of pain processing still are. Your brain is extracting the emotional meaning of the other person's pain without actually feeling the physical sensation itself. Empathy is also modulated by many factors, including your relationship with the person, your cultural background, and your own emotional state. You tend to empathize more with people who are similar to you or who belong to your social group. This in-group empathy bias is reflected in brain activity. The empathic response is stronger when you're watching a member of your own group in pain compared to a member of an outgroup. This bias can contribute to social prejudice and conflict, but it can also be reduced through interventions that promote perspective taking and contact with out-group members. The neural basis of empathy is plastic, shaped by your experiences and intentions. Oxytocin plays a crucial role in social bonding and empathy. Oxytocin is released during positive social interactions, and it enhances the activity of brain regions involved in social cognition and empathy. It makes you more attuned to others' emotions, more trusting, and more motivated to bond. Mothers have surges of oxytocin during childbirth and breastfeeding, which promotes maternal bonding and caregiving. Couples have increases in oxytocin during intimate moments, which strengthens their romantic bond. Even hugging a friend or petting a dog can release oxytocin, creating feelings of warmth and connection. Oxytocin doesn't just make you feel good. It changes how your brain processes social information, making faces more appealing, reducing amygdala responses to threat, and increasing trust and cooperation. But oxytocin is not a simple love hormone that makes everyone feel warm and fuzzy toward everyone else. Its effects are context-dependent and socially selective. Oxytocin tends to increase positive feelings toward members of your in-group, people you already feel connected to, but it can sometimes increase negative feelings or defensive aggression toward perceived threats to your group. In this way, oxytocin is more about strengthening social bonds within groups than about promoting universal love. It's a bonding hormone, and bonding inherently involves boundaries, distinguishing who's part of your circle and who's outside it. The social nature of emotions extends beyond empathy to emotional communication. Humans are extraordinarily expressive. We communicate our emotions through facial expressions, body language, tone of voice, and subtle behavioral cues. We're remarkably good at reading these signals in others. This ability to send and receive emotional signals is crucial for social coordination. It allows us to know when someone is angry and we should back off, when someone is sad and needs comfort, when someone is happy and wants to share their joy. The brain regions involved in recognizing emotions in others overlap with those involved in experiencing emotions yourself. The amygdala, for instance, is crucial for recognizing fear in other people's faces. People with amygdala damage often can't accurately identify fearful expressions, even though they can recognize other expressions and identify faces. The amygdala seems to be specifically tuned to fear signals, perhaps because recognizing fear in others can alert you to danger even if you haven't directly encountered the threat yourself. The fusiform face area, a region in the temporal lobe specialized for face recognition, works together with the amygdala and other limbic structures to process emotional expressions. This system is so sensitive that it can detect emotional expressions even when they're presented too briefly for conscious awareness. In experiments where fearful faces are flashed for just a few milliseconds and then immediately masked by a neutral face, people report seeing only the neutral face, but their amygdalae still respond to the fearful expression. This shows that emotional processing can occur outside of conscious awareness, influencing your feelings and behavior even when you're not aware of what you've seen. All of these systems, the limbic structures that generate emotions, the prefrontal regions that regulate them, the neurotransmitters that modulate them, the social circuits that share them, work together to create your emotional life. Emotions are not simple, primitive reactions. They're sophisticated, flexible, context-sensitive phenomena that arise from the coordinated activity of multiple brain systems. They're shaped by your genes, your experiences, your culture, and your current environment. They change over time, influenced by learning, aging, and the plasticity of your brain, and they're essential, not just for feeling it good or bad, but for making decisions, forming memories, building relationships and navigating the complexity of human existence. Understanding the neuroscience of emotions doesn't diminish their beauty or their importance. If anything, it deepens our appreciation for how remarkable emotions are. The fact that a collection of cells, communicating through electrical and chemical signals, can create the subjective experience of joy or sorrow or love is one of the most profound mysteries in science. We've made enormous progress in mapping the brain systems involved in emotion, in identifying the key structures and circuits and chemicals. But we're still far from fully understanding how physical processes in the brain give rise to the felt quality of experience, the what-it's-likeness of feeling. This is the hard problem of consciousness, and emotions are at the heart of it. What we do know is that emotions are deeply embodied. They're not just in your brain, but in your whole body. When you feel afraid, it's not just your amygdala firing. It's your heart pounding, your muscles tensing, your breathing quickening. When you feel sad, it's the heaviness in your chest, the tears welling up, the slump in your shoulders. The brain and body are in constant communication through the autonomic nervous system and the hormonal system, and emotional states involve changes throughout this entire system. Some theories of emotion, like the James Lange theory, propose that emotions actually begin with bodily changes and that what you experience as an emotion is your brain's interpretation of these bodily states. You don't run because you're afraid. You're afraid because you run. Modern versions of this theory, like Antonio Damasio's somatic marker hypothesis, propose that emotions arise from the brain's representation of bodily states, and that these embodied signals are crucial for decision-making and consciousness. The interoceptive system, your brain's representation of your internal bodily state, plays a crucial role in emotional experience. The insula is a key hub of interoception, receiving signals from your heart, lungs, gut, and other internal organs, and integrating them into a coherent sense of how your body feels. People who are more interoceptively aware, who are more in tune with their bodily sensations, tend to experience emotions more intensely. And practices that enhance interoceptive awareness, like mindfulness meditation and certain forms of yoga, can change how you experience emotions, making you more aware of the bodily components of feelings and better able to regulate them. As you lie here tonight, resting, drifting towards sleep, all of these systems in your brain are still active, still processing, still creating your emotional experience. Your amygdala is monitoring for threats, but finding none, it's quiet. Your prefrontal cortex is relaxed, not needing to exert much regulatory control. Your neurotransmitter systems are shifting into sleep mode, serotonin winding down, dopamine quieting, as your brain prepares for the restorative work of sleep. Your hippocampus is busy consolidating the memories of the day, deciding which experiences are important enough to keep and which can fade. And through it all, your emotions, those fleeting feelings that have colored your day, are being woven into the fabric of your memory, shaping who you are and who you'll become. Emotions are not obstacles to clear thinking or signs of weakness. They're intelligent responses to your environment, shaped by millions of years of evolution and by your own unique history. They motivate you, guide your decisions, connect you to others, and make your experiences meaningful. They're what make you human, what give your life its richness and depth. And every emotion you feel, from the most subtle flutter of contentment to the most overwhelming surge of love or grief, is the result of your brain's extraordinary ability to create subjective experience from the dance of neurons and chemicals and electrical impulses. You are your emotions, in a very real sense. Not because emotions control you or define you completely, but because they're inseparable from your experience of being alive. Your emotional brain is part of your thinking brain, your social brain, your deciding brain. They're all one system, one unified whole, working together to navigate the world and create the experience of being you. And tonight, as you rest, you can appreciate the remarkable complexity of that system, the beautiful machinery inside your skull that lets you feel everything you feel, and know that it's all working quietly, constantly, to keep you alive, to help you thrive, to make you capable of experiencing this one wild and precious life. If you've made it this far, and you're still awake, thank you for staying with me on this journey through the emotional brain. If these gentle explorations into the science of how we feel have helped you relax, or given you something interesting to think about as you drift off, please consider liking this video and subscribing to the channel. It helps these videos reach more people who might find comfort and curiosity in understanding the brain. And if you would like to continue learning, there are more videos waiting for you, more quiet journeys into the wonders of science and the mysteries of the mind. For now, let yourself rest. Let the thoughts settle like snow falling softly. Let your breathing slow and deepen. Let your body relax completely, every muscle releasing its tension, every part of you becoming heavy and still. Your brain will keep working through the night, processing the day, preparing for tomorrow, creating dreams from the fragments of memory and emotion. But you don't need to think about any of that now. You've learned enough for tonight. You've traveled through the limbic system, visited the amygdala and hippocampus, explored the prefrontal cortex, and traced the pathways of neurotransmitters through your brain. You've seen how emotions are created, how they're remembered, how they're regulated, and how they connect you to others. And now you know that every feeling you've ever felt, every moment of joy or sorrow or fear or love, is a testament to the incredible complexity of your brain, to the billions of neurons working in concert to create the experience of being alive. You are a feeling being, an emotional creature, and that's not a weakness or a flaw. It's your greatest strength, your deepest connection to yourself and to the world around you. Sleep well. Dream gently. And may your emotional brain, that ancient, beautiful, endlessly fascinating part of you, guide you through restful sleep and into tomorrow with wisdom, resilience, and grace. Good night.